which is Ilenna Jones, and she is a PhD student from the University of Pennsylvania in Conrad Cording's lab. And she'll be talking about if single neurons can solve MNIST.
Hey, thank you so much, Stephen. I'll share my screen. All right. Can everyone see that? All right, cool. So my name is Ilenna Jones. I'm a PhD candidate in Professor Conrad Cording's lab, and my talk will be about can single neurons solve MNIST, the computational power of biological dendritic trees. Oop. Since the neuron doctrine, we've asked, what can a neuron do? Do is a rather vague term, but we can define it. We can define it in terms of role. So what do neurons do to mediate behaviors? If you remove the neuron and the behavior stops, does that mean that that neuron is a neuron of that behavior, and so on? We also could define do in terms of computational capability, wherein we would consider a neuron as an input-output device, where the input is received in its dendritic tree, integrated into the soma, and producing a binary, or at least how I'll be talking about it, a binary output of the presence or absence of an action potential. I'm not the first to approach a neuron as an input-output device. In 1943, McCulloch and Pitts made the McCulloch and Pitts neuron, which was a linear weighted sum of the neuron's inputs put through a thresholding nonlinearity, producing a binary output. And if you hooked a bunch of those together, you could do logic functions. And if you fast forward about 70 years, you reach a very key artificial neural network, ImageNet, by Khrushchevsky et al. 2012, where they were able to hook up a whole bunch of neurons, thousands of neurons, and these neurons, which are actually quite familiar, they're linear weighted sums of their inputs put through some form of output nonlinearity. And if you hook them all up, you can do a high dimensional image classification, where you're able to identify the objects within the images that are present in these high dimensional, high resolution pictures. Both McCulloch and Pitts neurons and artificial neural network neurons are linear weighted sums of inputs mapped through an output nonlinearity. This follows the point neuron assumption, wherein that assumes the dendrites are linear. However, dendrites are nonlinear. They can do logical operations, coincidence detection, low-pass filtering, and other things. Point neuron models actually might be grossly underestimating what neurons could do. So how do we ask what neurons can do? A traditional way is the empirical neuron modeling procedure. It's a very common procedure, very typical, where you would take an input current that you know, and you input that current to a real neuron and record its empirical output. And then you take that same input current that you know, put it into a mathematical model, neuron model of your choice, and then observe what the output is. And what you want is for the output to match the original empirical recording. What you do in order to make it match is you do some form of optimization or training of the neuron model. And when that neuron model, given some input, produces an output that matches the empirical recording, you have a good neuron model. And this can be used to test counterfactuals and other conditions that you may not have tested in the original neuron, and so on. It's very useful. However, the other method of how do we ask what neurons can do that I'm proposing is led by this question. Like with artificial neural networks, can we test a neuron's ability to perform tasks? In this task-based neuron modeling procedure, I put them side by side. It's the input and output that are different. And I will say clearly now that the task that I have in mind is a binary classification task. Binary, and because neurons, as far as I'm considering them, produce a binary output. And I also have in mind for my neuron model that I'm going to ask questions about the dendrites and to be able to ask questions about the dendrites, I need to have higher dimensional, multidimensional input. So I'd have this dendritic task input, which I'll define soon, and then the output is a correct binary classification. And the reasons why I do this is to take a principled approach as to setting what the dendritic inputs are, and so that I can quantify the neuron model's performance via an accuracy measure, either it is correct or incorrect. So what did I mean by binary classification? I turned to machine learning benchmark tasks. The first one of which I will talk about is MNIST, which is a handwritten number data set, pictures of numbers that are handwritten that are 0 through 9, so there are 10 classes. I have 2 classes here that I choose for my binary classification task, and I've chosen them by taking a very simple linear classifier and seeing which ones are the least linearly separable. And another way of saying this is which 2 classes are the most confused when you put them side by side. And 3 and 5 were the most confused pair. So I have I have an input and output function. The input being an image of a number that is, when padded, a 1024 dimensional, each pixel counts as a dimension, image. And the output, which is the target label, 3 and 5, or what I already assigned them to is 0 and 1. So what about my neuron model? I have my input, I have my output. What about my neuron model? And what I suggest for my neuron model is the one tree. And I'll make an aside here. Choosing your neuron model basically allows you to ask particular kinds of questions and hypotheses. I directly choose this neuron model in order to ask questions about a property of dendritic morphology. And that property I'll talk about now. So if you look at this cartoon of a neuron, I have these inputs, 1, 2, 3, 4, assumed to be only on the terminal ends of the dendritic tree. You'll see that in order for those inputs to reach the soma to be integrated, it needs to travel through this very strict binary tree structure. And so I wanted to ask, what about, does this binary tree structure have some sort of impact on computation? So I turned to deep neural networks, and I made a network that has this dendritic sparsity constraint and called it a one tree. And it's comprised of nonlinear nodes representing compartments of a dendritic tree. And so I want to compare the performance of this one tree on my binary classification task to a linear classifier that is a proxy for a linear point neuron model, and a densely connected, very much the opposite of a sparsely connected, but a densely connected, fully connected neural network that has the same number of parameters. So it's the same exact, well, not exact, but the same size of the one tree. So this is a very busy slide, but I'll direct you to A. And so what we have is the one tree, we have accuracy on the y-axis, and we have the one tree, the linear classifier, and the fully connected network. And so the one tree performs better than the linear classifier. The linear classifier can be kind of seen as a lower bound, but it performs worse than the densely connected, the densely connected network, or the fully connected network. And what you might conclude from this is that perhaps the sparsely connected nature of a neuron might limit the computations that a neuron is capable of compared to that of a fully connected network, theoretically. So this is all on deep networks. So of course, I'll have a slide talking about the limitations and assumptions of this later on, but this is the conclusion that I'll make. Oh, and we repeated this in other data sets, and most of which repeated what we found. And I'm not going to go too much further because I want to get on with time. So what else could impact single neuron computation? Neurons have thousands of synapses, and presynaptic axons synapse on average 4 times per postsynaptic neuron. This means that if you have a postsynaptic neuron, on average, you'll have 4 repeated inputs from the same presynaptic neuron onto that postsynaptic neuron. In vitro multisynaptic batons onto the same dendrite increases approximately 6-fold after LTP induction protocol. What this basically means, with the takeaway from this, is that learning may impact the replication of synapses. And so this leads us to the hypothesis: repeated synapses may also impact single neuron computation. So we try to test this hypothesis using the one tree. So if you look at this cartoon of a neuron here, I have these terminal inputs, 1, 2, 3, 4 again. And what if I repeated those inputs to other subtrees of this dendritic tree? That leads me to this structure here, the K tree, where you have these subtrees with independent weights receiving the same exact inputs across the whole model. So in this way, I'm repeating the number of inputs, and I'm also increasing the size of the tree. So how do I deal with the fact that I'm increasing the size of the tree in my apparatus? I try to modify the fully connected network to match the K-tree parameter size. So using these expressions where n is equal to the number of inputs to either the K-tree or the fully connected network, and h is the number of hidden layer nodes, and k is equal to the number of subtrees. If h is equal to 2k, then the amount of parameters is approximately equal. So another busy slide, I'll direct you to 8 again. So the y-axis is accuracy. The x-axis is the value k, so 1, 2, 4, 8, 16, and 32 determines the size of each of the respective models. And here I have, no, I lost my mouse. Here I have labeled the linear discriminant analysis, or the linear classifier, as a point of comparison. And you can see that in gray, you have the fully connected network, again, above the K-tree at the one tree level and stays consistent throughout. But you can see that the gap between the fully connected network and the K-tree seems to narrow as you increase K. And actually, if you look at fashion MNIST, eMNIST, in the USPS data set, by the time you get to the K equals 32, that gap, it becomes statistically indistinguishable. They cannot be statistically significantly different from each other using t-test. And this is not necessarily the case for some other data sets. And CFAR10 was weird, so the fully connected network performance actually went down, making it so that the K-tree performance was significantly higher. But that is very odd, and I'm not sure how to explain that. So discussion and takeaways. This is the limitations and assumption slide. So the input-output function, or a 1D vectorized image classification task, puts our neuron model out of brain context. It's not likely that you'll find this kind of task in the brain. Also, our model is an abstract model of a real neuron. The neurons are not feed-forward. Synapse location is not limited to the terminals of the dendritic tree, and our model is receiving real valued inputs, not binary inputs. But knowing all of these things that are biological properties of a neuron model that one might want are actually next steps in what we can do to make this model even closer to a neuron and be able to test each of these properties. We can test recurrence, we can test synapse location, we can test binary inputs. But for now, the takeaway is that our results provide inputs on dendritic structure constraints and repeated inputs. This is a special case of sparse neural network. This sparse network can perform at the level of a dense network using nearly the same amount of free parameters. And future work would be to compare K-tree sparse network to other kinds of sparse networks, such as that in the lottery ticket hypothesis paper. This is also a relatively new approach. Considering bringing deep learning approaches toward neuron modeling, we judge neuron model performance instead of traditionally trying to match neuron physiology data. Also, I will acknowledge that some of the work coming out of Aydin Segev's lab also is along these lines, and I would definitely recommend checking them out. Our results suggest that repeated inputs to a dendritic tree could improve its ability to perform complex computations, and some future work would be to introduce more biological constraints and train these more biophysically realistic neuron models to do image classification. And with that whirlwind presentation, I thank everyone here. I thank the Cording Lab. I thank my advisor, Conrad Cording, the Neuroscience Graduate Group, and the University of Pennsylvania. Are there any questions?
Awesome. This is really interesting. We do have some questions. Let me get to them. So Daniel asks, to what extent are the electrophysiological processes at the branchings you model here observed to be plastic in the brain, not just the synapses?
Yeah, so I'm sorry, I'm like speedily running through these slides. I know people hate that. So the branchings are allowed to be plastic. I trained this model using a very biologically implausible process, the backpropagation algorithm. I do not use any neuroplasticity rules that have found in biology. That would be another way of trying to get this closer to what is biophysically realistic. Think of my work as kind of a first step at building this kind of, reference of, oh, this definitely has some impact on computation. That maybe not. This does, and so on. Sorry.
Awesome. All right. I think now we're gonna move on to our next talk.
Thank you so much.
Thanks again, Ilenna. So for our final speaker, we have Jorgen, and he is a postdoctoral associate at MIT in the lab of Michael Fee. And he'll be talking to us about a connectomic substrate of credit assignment in basal ganglia reinforcement learning.
Thank you so much for this introduction. So let's start the presentation. This works, right? So yeah, I mean, here in the background, you can basically see already that I'll talk about a lot of data. So about a basal or several basal ganglia connectomes, and I basically want to see what we can learn from this like static data about credit assignment in reinforcement learning. So let me first talk about what I mean with credit assignment. Well, this is actually a very general problem, as you know. So basically, when you want to optimize